Hello, everyone. This is testosterone signaling of the male fetus, Period 1 AP Biology, video number 215 by Arabella Kim and Lizzie Friedberg, narrated by Lizzie Friedberg. Let's get started. Testosterone is a hydrophobic signaling molecule produced by the testes before and after birth. Testosterone is an androgen, a class of hormones responsible for male sexual characteristics and normal reproductive function. It binds intracellular receptor proteins called androgen receptor proteins on target cells, and once activated, the receptor interacts with DNA to turn on genes coding for proteins involved in normal male development, thus playing an important role in the organization/sexual differentiation of the brain and masculinizing the body. To reiterate, the signaling molecule testosterone is a hydrophobic signaling molecule, meaning that it can diffuse in and out of cells through the cell membrane. Testosterone binds to proteins known as androgen receptors. Androgen receptors primarily serve as DNA, binding transcription factors that regulate gene expression. Androgen receptors are intracellular receptors, meaning that they're found within the cell. In the case of AR receptors, within the cytoplasm. When intracellular receptor proteins are activated, they cause changes in gene expression. In other words, they can turn genes on or off. Some advantages to this type of receptor protein would be that their responses are sustained over time and that they could produce long-term changes in cellular activity. However, some disadvantages would be that because the response to hydrophobic signaling molecules is a change in gene expression rather than enzyme activation in other proteins, the response to hydrophobic signaling molecules is relatively slow to start. In general terms, the pathway for a process involving an intracellular receptor protein would involve the release of a signaling molecule from a signaling cell, followed by said signaling molecule crossing the plasma membrane and binding to a receptor within the target cell. The receptor will activate, and the activated receptor will trigger a signal transduction pathway, which will ultimately result in a cellular response. General players involved within this particular intracellular response, that is, testosterone signaling within the male fetus, include HCG, human chorionic gonadotropin, which is a hormone produced by the placenta during pregnancy. HCG does not only support said pregnancy, but also can help stimulate fetal testosterone production. Testosterone itself acts as a signaling molecule, which works to bind to androgen protein receptors, one of the other players. Additionally, later on in the process, androgen receptors will recruit various co-activator and co-repressor proteins such as NCOR, SMRT, HGAC1, and HGAC2. Within a male fetus, HCG transported through the placenta can act as a stimulus, encouraging the production of the signaling molecule testosterone within the testes. Testosterone not yet attached to proteins is known as free testosterone. To initiate androgen action, free testosterone enters the cell and crosses the plasma membrane, a movement that's enabled by testosterone's hydrophobic nature. Once within the cell's cytoplasm, testosterone binds to the androgen receptor protein, AR, which becomes activated and undergoes an alteration in conformation, shape, or structure, which allows it to be freed from heat shock proteins. Heat shock proteins help maintain the unbound AR in a shape competent to bind ligands. Once bound to testosterone and having experienced active conformation, AR is then able to translocate to the nucleus, where it binds to specific DNA sequences called androgen response elements, ARE, after undergoing dimerization and phosphorylation. AR binding to ARE permits for the recruitment of co-activator and co-repressor proteins, which alters the expression of genes. This bond modulates gene transcription, which leads to the transcription of mRNA, which exits the nucleus and undergoes translation, which ultimately results in the synthesis of proteins, moderating the effects of testosterone on target tissues. In a male fetal case, this will be the development of typically masculine physical characteristics like body hair, strength, muscle mass, and the development of male-specific organs. In the case that a body needs to stop the effects of testosterone, for example, if testosterone concentration is especially high, it breaks down the hormone and breaks the androgen receptor. Bond testosterone degradation occurs when enzymes break down testosterone molecules into smaller components, helping in the regulation of hormone levels and preventing overstimulation of cells. This process ensures proper hormonal balance and avoids potential health complications associated with excessive testosterone activity. Alternatively, the ligand withdraws or degrades. Post-translational modifications to the androgen receptor, such as dephosphorylation, referring to the removal of a phosphate group from a protein or molecule, typically by enzymes called phosphates, occurs. Dephosphorylation can modify, or in the case of androgen protein receptors, deactivate the protein, turning off the signaling pathway and leading to the AR returning to its original state in the cytoplasm, waiting for another testosterone to bind to. Testosterone signaling pathways are slow and sustained, meaning that it could take a longer period of time before termination hits and the signaling molecule disassociates from the receptor protein. Testosterone signaling pathways are utilized throughout an individual's lives, and the more dramatic effects of the negative feedback loop, creating the first termination example we discussed, is much less common than the second discussed process. The antagonist that we're going to discuss is enzalutamide, an exogenous antagonist frequently utilized to combat conditions such as prostate cancer. Enzalutamide is a competitive inhibitor for testosterone. In other words, it sits in the binding site of androgen receptors, where testosterone should be. As a result, it blocks the binding of testosterone to androgen receptors, and by proxy, prevents the rest of the process from occurring. This means that by proxy, enzalutamide inhibits conformation, binding nuclear translocation of the activated androgen receptor, and impairs the binding of activated androgen receptors with DNA. As a final result, the final intent of testosterone signaling to alter gene expression doesn't occur. On a larger level, as an anti-androgen, one might assume a certain level of enzalutamides administered to male fetuses would result in difficulty expressing male traits. In other words, fetal exposure to anti-androgens, such as enzalutamides, would disrupt male sexual differentiation. Thank you guys for listening, and we hope that you enjoyed.